I'm doing things in the lab today that I never envisioned that I would do when I started the lab 14 years ago. I'm Castro. I'm a professor in the Department of Mechanical and Aerospace Engineering. My lab focuses in DNA nanotechnology for biological applications. Our research focuses on making devices and tools and robots that can interface with biological systems. Rather than thinking about, say, a human or an animal, we're often thinking about biological systems in terms of cells and molecules. And so we're very interested on how do we make devices at that scale that can interface and operate in those systems. We're using DNA in a very different way than its natural biological function. We basically use it to make mechanical devices and structures and sensors and actuators. What we're really interested in is getting those into biological systems and using those to measure what's going on, but also to influence processes that are going on. So maybe we can even do things like try to fight certain mechanisms of disease. Our general approach is a lot like what you might think about when an engineer is trying to develop an engine or a bridge or something like that, right? When you do that, in order to optimize the design, you often might put in things like sensors, force sensors, pressure sensors, temperature sensors, just to understand how the system is operating, and then of course tweak design parameters, right, to optimize things like efficiency or power generation. And we kind of approach this design problem of understanding how cells and molecules work in a very similar way. Fabrication is different than a typical engineering fabrication process. It's a lot of pipetting. We take something like 200 different DNA strands, pipette those all together into a tube, and then we run that through a self-assembly reaction. And that involves basically heating the solution up to a certain temperature so that all the DNA strands are melted, and then we cool it down slowly so that all those DNA strands can bind together and find those sequence-specific interactions to form the compact structure that we're trying to make. All you need to know about how DNA self-assembles is that there's 4 different building blocks or nucleotides: a G, a T, and a C. An A base binds to a T base, and a G base binds to a C base. And that turns out to be very powerful because if we have some sequence of DNA, say GAT, CCA, something like that, we could design another sequence that should bind to that first strand. And that allows us to program how these DNA strands will self-assemble into nanostructures. Our lab actually published the first example of folding a gene into these DNA origami nanostructures that we work with, but then also being able to deliver that gene to a cell and have that cell express the genetic information. And we think that has potential to address some really important challenges in gene delivery, because it is quite hard to take a very large gene and get it inside of a cell even in the first place so that it can be expressed. Currently, what people often do is just take pictures of the system, right? Lots of fluorescence imaging to see what are the structures, where are the cells, where are they moving, but it's very hard to get real-time information. So that's where we think these tools can be very useful, is really getting real-time information on what are the forces that the cell is experiencing, what are the molecules that the cell is interacting with. And now we're moving on to what we're really excited about, is how do we get them to do stuff. We're collaborating with a leukemia-focused research group in our Comprehensive Cancer Center to develop therapies that we can put molecules on these DNA nanostructures that are loaded with drugs, basically to get them to interact specifically with the leukemia cell versus a healthy cell. We're hoping to move that into, say, mouse model testing to see, can we really then use that to, in a targeted way, deliver a drug.